So my name's Tim. I've come over from Cardiff University at the moment. I'm in the final year of a PhD where I've been trying to see if we can do trying to find any pattern matching within the field of schizophrenia genomics. Okay, so I'm going to start this talk off with a quick personal request, because the usual audience when I'm giving talks are experts in biology, psychiatry, and very few of them have little, if not no, experience of machine learning or coding. So if at any point anyone is feeling like this during my talk, please, if there's any problems, please let me know. If it's really bad, maybe let me know outside so it's not on camera and I can eat biscuits. Okay. Thank you. Right, so I'm going to give a very quick background into schizophrenia and its genetics. It's a debilitating psychiatric condition that has a lifetime prevalence rate of about 0.7 to 1% of the population. Now, it's associated with a whole variety of different symptoms, some of which are quite commonly known amongst the general population, but you also have those that are not so well known, often those coming under this negative symptoms that we have here. So, I found this on the internet the other week, and I found it's quite an interesting image because when I'm talking to the clinicians that work in my department, you realise how awful this disorder is for people, because they really are trapped inside torment within their own minds, and for some of them it's as if they can't get out of their own nightmares. And it's very easy for me to sit down, I get bogged down by looking at numbers and trying to find pattern matching, and it's really easy to forget actually what we're trying to do, which is to get rid of people going through things like this. So as for the genetics, it's what makes the genetics of schizophrenia interesting but also difficult is that you don't have like a single tag. There's no single biomarker or mutation that you can have that will say this will give you schizophrenia. If you contrast that with, say, something like Huntington's disease. But what instead is that you have these small sort of cumulative contributions from all over the genome, and these are often very common mutations that all of us here will have some of the mutations, but it's a case of there's certain patterns that you can have that again won't determine whether you'll get it, but they can increase your probability of getting it. So that's what makes this really quite interesting to research. So if we look at the sort of mutations that I deal with, are these common mutations that we call SNPs. So just to give an example here, this is where, because on the most part, all of our DNA, we share a lot of our DNA. We share a lot of DNA with our closest ancestors, of the primates. And I think it's something like 2/3 of our DNA we share with a banana. So there's a lot that we have in common. But there are these particular places where they've spotted these single nucleotide base changes in the genome. And so here you've got an example in this diagram here, where for that section of DNA it's all the same apart from that one point. Now what the diagram here shows is that there's 3 different types at this point. On the most part, what we're looking at is that there's normally one or the other. And a little section of DNA that has these mutations is what we refer to as an allele. So this is a triallelic mutation, but for the most part it's a biallelic that we're looking at. As an example here of one of the mutations that has been found to be associated with schizophrenia on chromosome 1. So just to point out that because we have double-stranded DNA, you can either have none of these mutations, one of these mutations, or if you get it on both of these strands, then you have 2 of these mutations. So that will be used in the calculations later. So one of the tools in the armory in psychiatric genetics is a Genome Wide Association test. And what this does is it looks at the distribution of the major alleles, so these are those base pairs that are more commonly seen in the population, with the minor alleles across cases and controls. So you get a 2x2 contingency table for each mutation. And then what you do is you calculate an odds ratio from this, the distribution amongst the cases and controls, and you can also get a p-value from a chi-squared test. Now, when we enter into these things, there are millions of these mutations that get put in, so there is need of multiple comparison correction. You don't need to do it for every single one because there is believed that there are 1 million independent sections of the genome. And when I say independent, what I mean is the crossover recombination that happens during reproduction between your chromosomes. There are certain breakpoints on these, and the assumption is that this results in 1 million sort of sections of the genome. So we're effectively doing 1 million independent tests. So what they do is they correct for this with a Bonferroni correction, factor of 1 million, which takes the threshold value down to p times, which equals 5 times 10 to the minus 8. Just a quick note that if you're dealing with ratio data, which is distributed about 1, it's often nice to take the logarithm of it, because the odds ratio is not evenly distributed about 1. If you take the logarithm, that makes it evenly distributed about 0. So that's what we put into the calculation. So this slide has been going around pretty much every single psychiatric genetics conference. It was a paper that was a bit of a name drop for Nature there. So they made it to Nature, and what they did was an enormous consortium study of schizophrenia sufferers from labs all over the world, of which Cardiff was one. And what they do is they did this, took all of these and they looked at all of the different mutations, and what they found was that there were 128 of these SNPs that were significantly associated with schizophrenia at this stringent level. And what we're seeing here is something called a Manhattan plot for self-explanatory reasons. And what we've got is you take the negative log 10 of the p-value, because often these p-values are very crammed, very close to 0. So you take the negative log 10 of it, which stretches it out so that you can see the differences more clearly. And what we can see is anything that gets past this red line meets the criteria. So just a point, we're interested in those that are the little diamonds at the top. The green ones, they've also sort of met the criteria, but they're within these chunks of genome that I was talking about. So basically, if you've got one of those, you've got the diamond one, and it's the diamond ones that we're interested in. So the aim of my study was, I was looking at a subsection, actually some of them that were genotyped at Cardiff, and these are people that have treatment-resistant schizophrenia. First of all, to get a smaller sample, and secondly, to see if there was something we could drill down about these particular sufferers. Is this a sort of sub-phenotype within schizophrenia? And these are often some of the most, from the clinicians tell me, these are people that really suffer from the disorder. They've been on medication that has failed for at least 2 years, I believe, and they're receiving this drug. It's this sort of drug of last resort called clozapine. There is an unequal balance of cases and controls, but I'll show how Scikit Learn really nicely deals with that later. So what I do is I enter the number of the minor allele counts, I enter that into the support vector machine model, and I compare it with the traditional method that's used in the field, which is to create a polygenic score. So just quickly go over that. What they do is they take these counts, 0, 1 or 2, take them for all the mutations, multiply that or weight it by the log odds ratio that they've got from the GWAS. Now importantly, that has got to be a GWAS with these individuals removed, and they've done that. That's fine, because you can't use their case control status to make a metric which is then in turn used to predict their case control status. So that has been covered by the consortium group. So you get, with that, you get a sort of a weighted allele counts for these people, and you take the mean of it, and that gives you a single risk score per person, which they call the polygenic score. And you can then look at this with logistic regression to see if you can predict case control status. They've said its predictive accuracy is very weak. It cannot be used for prediction in schizophrenia. In fact, because of the low numbers of people that suffer from schizophrenia, if you want a classification model, just point at someone and say, you haven't got schizophrenia. Because in that case, you're going to be right 99% of the time. And the accuracy of this is nowhere near to that. So there is room for people to try and improve on it, which is why I've been working on in this field. I've been using the support vector machines because what it does is if you're taking a sort of a sum of all of these features like the polygenic score does, it doesn't take into account any nonlinear effects that could be happening. So that's why I've used linear and nonlinear kernels for the support vector machine. So if anyone's not familiar with it, I'm just going to a very quick recap of what they're trying to do. This is a simple 2-feature model here. So what we've got, if you've got these 2 different classes, and a support vector machine is trying to find the optimum decision boundary between them. This is all like developed from pictures on Scikit Learn's documentation, which is as good as their software. So this is the optimum hyperplane. What does it mean by optimum? What it's doing is it's maximizing the distance between these dotted lines. And it's solved by it's a non-trivial quadratic programming tool that does this. But interestingly, what it does is it uses these that are, if you can see it, some of them that are closer are actually on the dotted line. They're circled, and they're referred to as the support vectors. So it's the comparisons between these points that get used to make this hyperplane. So when you're building these models, you'll hear about parameter tuning that takes place, and this is an example of why. If you've got a point here, which it could have been measured correctly, is it blue? But it looks as if it really should be. Okay, can everyone see this? Is it clear? it looks as if it really should belong in the other group. So you can set this cost parameter called C. Now you can say, no, get it right, and it'll do something like this. So it'll do what you're telling it to. But a better approach might be to do something like this, which means that you're letting this one go. You're saying, okay, fine, let that one go. But this model, we've got our nice large margin back. This will probably, hopefully, be more generalizable to new data, which is the whole point of building these models. And this also shows that a support vector machine is capable of building a soft margin classifier. It doesn't need to get everything right, so it can solve problems that aren't completely linearly separable. That being said, there are cases where it's really not going to work. And this is sort of a variant of a classic example called the exclusive OR example, where if you've got high values in one or the other feature, then you'll get the disorder. But if you've got none or you've got both, then you're not going to have it. So you could maybe one of them cancels the other one out, for example. Bit of a toy example, but here we go. Any attempt at a linear boundary is just not going to work with this. So what we do is we do use a kernel function. And that's essentially, it looks at the relationship between the support vectors in a different way, and we get this. We're now back to 100% accuracy. And you might hear that people say, oh, this problem is not linearly separable in 2 dimensions, but it's linearly separable in a higher dimension. And the way I visualize this is it works something like that. So now, if you're plotting the points, rather than plotting those points that were in orange onto a flat surface, they're now actually effectively, they're going to appear at the top of these mountains. So now if they're now there, you can then put a linear plane through them and you've got your linear separability back. Okay, so the next thing about machine learning is half the battle is finding out what exactly you're going to put into these models. Now, I could use the weighted, hello?
Do you mind if I ask a question about the way that you sort of produce the 3 dimensions from 2 dimensions? If it was the exclusive OR,
yeah,
it seems like that would seem a little bit odd. So, like, is there a way that you would choose?
Oh, that's just a way of how I'm visualizing when people say if you take it into higher dimensions. Because sometimes if you're talking about many, many dimensions, you can't visualize it, but that one you can. It's purely a visualization tool for it. So just going back to what I was going to enter into these models, I was thinking about entering the weighted allele counts like the polygenic score has, because that provides some important information. If you've got a mutation that's seen more in controls, it's going to have a positive value, and then negative value for the opposite if it's seen more in cases. But the problem here is that these allele counts are sparse. They have 0 values, because remember, you could have none of these mutations. And this really concerned me with the polynomial, and there's an example here of a quadratic kernel. So this is a polynomial with a degree 2, because what we're really interested in is the last term in that equation, which is an interaction term. Now, if anything you're entering in here is a 0, it's just going to knock that interaction term. You're not going to be measuring what you want. The RBF kernel's not so susceptible to this, but the only way to really check is to do a quick example. So I do an explicit XOR example I enter in here. I build the targets. I build the inputs and I build the targets. As you can see here, imagine that you have a sample per row. If they've got one or the other, then they've got the disorder in the targets, not for the other 2. And then you would never do this actually in machine learning, but I use the same model to predict the same inputs and just see how it deals with them. And it's done like this. Now, we're not expecting the linear one to get it right. It can't. The RBF does fine, as I thought, but the concern here, the polynomial one, is not getting it. So the alternative to that is let's scale the inputs. And what you do by scaling is done very easily in Scikit Learn. What you then do is you scale it so that for each feature, you have a mean of 0 and a variance of 1. And in fact, what it's done here is it's swapped out the zeros for a -1. If we do that, run it through, and both of the nonlinear kernels are performing as we would expect them to. So that made me decide I'm going to use scaled features. I'm not going to allow the zeros to go into it. So now that I've decided on the features, we now need to talk about the parameter search. And this is where it really made my life easier. I'll go on to show why. Because from some initial trials, I found that it was the lower values of C and this other one, gamma, which gets used for the nonlinear kernels. So it was the lower values. So what you can do is rather than giving explicit values in a grid search that you want it to look through, you can give Scikit-Learn probability distributions. So you say, sample some points from this probability distribution, carry out 4-fold cross-validation. Is everyone clear with cross-validation? How you test on, you hold some data out, then, okay. So anyway, so what you do is you pick some parameters from these distributions, 4-fold cross-validation, get the best score, and then you keep iterating that many times until you get what you think is the best combination of parameters. Sounds complicated? It is this easy in Scikit Learn. Where after you import some of the models, load in your information, then what you do, you build the CV model, the cross-validation model, build the support vector machine. The scaling has to be done in a cross-validation model as well. You have to do that in a pipeline scaling. So rather than scaling it all, because then the test information would have information on the scaling from the training data, what you do is you find out the mean and the variance of the training data, and then you apply that to the test data, but that's done very easily with this pipeline feature. Then you build the probability distributions, and then you run it through this model here, this randomized search CV, and it does everything for you. I actually wrote into a forum about this. Someone said use this, and it's that easy, and I was very happy about that. So just to show how well this has been thought out, it's really made my life so much easier. So let's go on to some results. Spoiler alert. Oh, sorry, there's one thing I forgot to point out here as well. This njobs equals -1. This is a really nice feature because it uses the joblib library, which I tamper with a little bit, but I more leave their implementation to it. What that says is use all the CPUs available. So when I send this off to a cluster, I can send it off to a node that's got 16 CPU cores. I can do that at night, and then it's all done then by the next morning. So it really is nice. You can just say use all the resources that you've got, and it's that simple to do it. The results. So ultimately, what I was hoping when I start the PhD, because you all think you're going to solve science, no. Unfortunately, we can't. And what I've also got here is, hang on, I'm just going to say one small thing here. This class weight equals balance. That's the thing that deals with the imbalance between the classes by applying a different cost function to each of them. So anyway, back to this. What I was going to point out, I had some extra data here that was actually done on a different genotyping chip. I initially kept it out for completely separate test data. It was performing similarly, so I entered it in as well. Now, unfortunately, the support vector machines are not doing as well as this polygenic score, and the even-valued polynomial kernels are completely bombing. If I've got some time, I'll show that I've done some simulations to find out why is this happening. This is indicative of main effects telling the whole story. If I've got time, I'll show that. But again, even when I've added these extra sample points, it's not really improving on the score. However, the story is a little bit different with this one. Again, I'm not solving schizophrenia, but the polygenic score is not improving at all when I add any of the extra features. And what they've used here is rather than just use those GWAS significance SNPs, I've relaxed the threshold. And I've taken it down to around 5,000. The reason it's 4,998 was because there was a nice p-value cutoff that I could use here. And I said, right, I wanted 5,000 maximum because after you've stripped the training sample down, I still wanted more samples than features that I was using. That was around 5,000. So I say, let in all of those sub-threshold SNPs. And what we're seeing here is, again, right, yep, the polygenic score is doing the best. This is the actual value that they got from that Nature paper, so I'm replicating that. But here, right, some of the nonlinear kernels are performing a bit better. The reason I'm using a mouse, I did have a pointer, but it doesn't work on these screens. So that's why I'm doing this. And also, all of the support vector machines are now benefiting considerably from this extra data. By the way, the metric is the receiver operating characteristic. Is everyone familiar with how that works? Okay, what it is what you do is imagine if you were to relax the boundaries of it. So if you were to move the support vector machine boundary, you move it and you look at the trade-off between false positives and true positives as you move it. So if it's completely chance, if they're randomly distributed, then you'll get a value of 0.5. 1 is perfect classification. So 0.65, don't think that's still, 0.7 is still pretty bad. So that's the reason why we're trying to improve on these things. So the question is, can we, if we really boost more samples, can we get more information coming into this? Now, I don't know. That's an open-ended question, because it could be the case that it asymptotes around 0.7. Nothing that we enter into it is going to be improved. But the alternative to that is, are we going to still see that with more and more people, these are going to creep up? The nonlinear kernels are going to do better, suggesting that there are interactions. We don't know. It's an open-ended question with that. But I still thought, well, my supervisor found this quite exciting, and we're talking about it next week. So the next thing is, can we get, because that's putting a lot of features into the model. That's 5,000 nearly 5,000 features going. These take a long time to run. Is there any way that we can get the signal from across the genome, but reduce the dimensionality in such a way, and yes we can, and this is where pandas really comes into its own, because what I was doing was putting these mutations in sort of functional groups that they come together. And the genes are the areas within the genome that code for proteins, and they also have been, from experiments, these are the people that work in labs with blue gloves, and I never go anywhere near them. Well, them, but I don't do it, not when they're working, because I've tried it and it's really not for me. There's nothing wrong with them. But anyway, they found that there are certain genes that are related in these functional sort of categories. And by looking at the different databases where they show the location of these genes, the gene sets, and the location of the mutations that we're using, there's really nice database functionality within pandas that you can prepare a data frame. This is an example data frame that I've got here. The values are actually, I don't know whether you can read the values because these screens are very small, but I've had to make them up because it's all confidential data. I've only got it for 2 samples here, so what you would have is you'd have 7,000 odd columns for the samples. But what's so good is that once you've got this is all the mutations mapped to all of their genes, all of their gene sets, and what we've got here are these sort of classes of gene sets. There's 6 of those. Pandas does it in 3 lines. You just use a group by functionality just as you would in SQL. And you just get the mean value. I dropped the reason why I dropped duplicates is because you can have gene regions that cross over with each other. So I eliminate that. And then you just group by on the gene set and take the mean. And that left me with 133 features, capturing. It didn't capture those mutations that fell in non-coding regions, but it was still, it was still a way of looking at how these are performing. Then we can enter these into the model. Some results. I focused on, at this point, using the linear model. The reason being is, as actually Ian showed in his previous talk, you can get information from the coefficients that are assigned to the features. Because in a support vector machine, it's actually describing a vector that's orthogonal to the decision plane. So when you project one onto the other, if you project a new feature onto that vector, if it crosses the decision plane, it changes class. If not, then it doesn't. So you can interpret these things. They're not black box. Once you stick a kernel on it, it does become more black box. So what I did here, I also used the multiple regression feature within Scikit Learn, and I was using this linear SVC. It's a slightly different optimization algorithm that they use. It's faster. It can only be used for linear kernels, but you can also apply this lasso penalty onto it, which means that if a feature is really considered to be doing nothing for the model, this can now assign it a value of 0 if needs be. Again, it would be really nice, wouldn't it? Are we improving on the polygenic score? No, science. But we can get, with the coefficient, we can get some interesting information out of this. And when I got this out, I was quite happy about this. Not immediately, I was like, oh, that's interesting. Read about it, and I was like, oh, this is really interesting, because these things at the top, FMRP, that describes a gene set called Fragile X mental retardation protein. And what this is, they're targets of this protein. This protein binds onto the product of the DNA called RNA from these genes that are listed here, and it transports these RNA from the nucleus of the cell to the synapses of the cell. So it has also, other lab work has shown that it's implicated in schizophrenia. So I then also used some other software called this MAGMA software, which is what's called gene set association analysis. And what that did, that found that this set at the top was the only one that was significantly associated with it. So I'm very happy to say that the support vector machines were basically singing from the same song sheet as other software. 2 ways to look at about this. I was quite happy saying, well, it's making sense. Of course, there's the attitude that one of my supervisors said, which was to come up to me and go, kind of means there's no real point in doing what you've been doing then. You can just use this other software. So yeah, there's 2 ways of looking at it. I'm going to go with my way, just so that it's still, obviously, because I've got to write something up. But of course we can go on from this, because what MAGMA can't do is it can't look for these nonlinear effects. If we use the kernel functions, are we going to show some interactions? Is this going to be really nice? No. It's also not going to be. I mean, we can't improve on this polygenic score thing here. I mean, but again, it's not to say that it's not worth doing. Maybe there's more information that we need to combine into this. And I remember when I started my PhD, someone said to me, don't worry about trying to solve too much. What you're doing is you're wanting to add a grain of sand to the beach that is human knowledge. And this could have been my grain of sand if that had been there, but instead, as is usual, I found this circling around social media the other week. And it sums it up, really. It sums it up. So, I'm glad that it's supporting other software. We haven't found any nonlinear effects, unfortunately. We might be able to see more if we have some more samples. That's what we always say. But we'll see from that. So, in conclusion, the support vector machines unfortunately didn't improve on the performance. There might be some interactions with the sub-threshold SNPs. But again, the gene set analysis did show it supported current research and other techniques. Now, how am I doing for time?
You've got probably 5 or 6 minutes to talk.
Okay, right, because what I wanted to do was, because we got these strange, if we go back to the this pattern that we're seeing, I was interested, what's causing this? How come these are bombing so badly? So I wrote, I'm going to show some code here, so I'm a little bit nervous about this. Don't scrutinise it too much. So what I've got here, if anyone can, it's very small on these screens. Can people, can you see it? So what I did here, what I wanted to do was basically say, okay, if we don't use the phenotype that is the case control status, if instead I make it, because I can say, let's make it from the main effects, or let's introduce interactions into the model, and then see if we know they're there, because we've made them, can the models find them? Because it would be really nice is the thing that you're thinking these things can find, can indeed they find them? That was why I wanted to do this. So I made this, I just wrote this quick script here that you take the inputs, you put it into the model, and I do choose to scale them so that you have the scaled input values. And this is just to make a phenotype. There's no machine learning taking place here. And then what I do, I can say, how many of the features do we want to use? Default is all of them. Now, importantly, how many interactions? Because the sort of thing we're looking for is, are there a subsection of these mutations that might be, their effect together is like, it's more than the sum of their parts. So we've got a nonlinear effect. So I say, only take 10% of them, make pairwise interactions. This penitence value might not be the best value, the best term to use here, but what it means is, if you meet the criteria for the main effects, this is your probability of having a phenotype. If the interactions make it, so I wanted this to be higher, because you want a strong effect of these interactions coming in. And you can additionally weight them, so because it performs a weighted average on these. Importantly with this is if you set the main weight or the interaction weight to 0, what you can say is only consider main effects, or only consider interactions, so on and so forth. Do some checks of what I'm putting in, and then I use this to basically make this phenotype, which is then returned at the base here. Anything? Yes, and it's really not, some of the functionality within NumPy, so we all need to thank Travis, and we'll get to see him talk in a few days, because it makes it so easy. You can do a weighted average to get these results, and it runs super quick as well. And then, this is the thing that basically drives it. You enter in the values that you want, and then you build this. This is all very much along the same lines as what I was doing before. And these are some of the results we get with the default that we have, where you've got 10% of the interaction, 10% of the features having a fairly strong effect on the performance. This polygenic score, I'm not too sure how to interpret this one, because I made it from the scaled allele counts rather than the actual weighted allele counts. So take that one with a pinch of salt. But what was nice to see is that the non-linear ones are behaving as we would expect them to behave. It's performing better than the linear in this case. Don't read into this too much, because it was just a quick way of making some phenotypes. With this one, I set the effect of the interactions to be far lower. I didn't weight them additionally, and I kept a very similar sort of penetrance level on them. And what we're beginning to see here are similar patterns to what we got in the real data, where the even polynomial kernels start dropping out. And with this one, I've set the interaction weight to be 0, so it's only going on main effects. And then what I was puzzled is, why are those polynomial even degrees really not performing? And when you make a feature purely based on main effects, or just to say how I decide whether it is or not, what I do is, if I've got all the values for the main effects, I take the value for the row. Sorry, I should have mentioned this. That's done here. if I got all of the main conditions, I take the value of the rows, which is an individual score, and if that score is greater than the global mean, I say, yes, they meet that criteria, and if it's below it, then they don't meet that criteria. Combine those two probabilities from the main effects and the interactions, just to see. It was just a way of trying to put something there and see if the machine learning could find it. And as I suspected, these were, if you main effects only, you do see a dropout for the even kernels. One thing that, I don't know if anyone knows about this and can help me with this, because one thing I was really quite puzzled at was when I set the value to be 3, so looking at three-way interactions, suddenly the linear has popped back into life again. The polygenic score hasn't, but this linear value has jumped up again. So if anyone knows why, I'm puzzled by that, but I would appreciate some help with that. So anyway, if we go back to my talk, I think that's it now. So thank you again, sums up my PhD. Thank you ever so much for listening, and thanks to my super. Thank you. Was that okay for time?
We have 15 minutes until lunch, so yeah, as many Q&As as you want. But just before we get started, someone on the Slack channels asked about slides, whether you have URL for these.
Right, yeah, I need to find out about them because this is the first time I've ever used an IPython notebook. They are HTML slides. I tried to sort of save them, Ian and I were talking about this yesterday, I tried to save them to PDF. When I find out how to do it, yes, I'll make them available. I will. So once I find out, yes, I'll go through the channels and do that.
The URL, can't you?
It's all locally. So yeah, I can see if I can do that. Can I talk to you about that afterwards and find out? Sorry. Okay. Okay.
We don't have a mic or anything, so just speak up and yeah, just pick your questioners and fire away.
Hi, Katie.
I have two questions.
Yep, that's fine.
So,
Thanks, Ian.
Copy number variants are very important in schizophrenia.
Yes, they are.
So, why aren't you considering those as well?
One of the reasons is, so if anyone, a copy number variant, what that is, because I'm looking at just a single mutation change on a point of DNA. What you can have is that you can have sections of DNA which for some individuals can actually be deleted completely, or you can have sections where it's just repeated, and you can have many repeats of these. A good example can be a deletion on chromosome 22. If you have that, you've got a very strong chance of getting schizophrenia. But they're also comorbid with other disorders, so that one is associated with severe intellectual disability. But to go back, the reason why I'm not using them is, yes, they've got a strong effect size on whether you get it, but such a tiny proportion of schizophrenia sufferers will actually have these mutations. And the problem is that when you're using machine learning, what you're asking it to do is look for commonalities of features amongst the sufferers that would have it. So I don't think tools like this would be that good for finding the CMVs, because what you're basically saying is, you're saying, oh look, this guy's got it, but then it looks at all of the other cases and go, well they haven't got it, so we won't rule it out. So that's right. But so you had a second part?
And so the SNPs that you're interested in were thrown up by Genome Wide Association Study.
Yes.
But they are not good at detecting interactions between variants, as far as I understand. So is it
No.
possible that they just missed a load of the SNPs that are actually of importance?
Actually, it wasn't a planning question. I'm glad she asked that, because there was something I forgot to mention here with this one, because I forgot to mention this, that when I decided which of the mutations fitted into genes, because what Katie mentioned then is they found these things called EQTLs, which are expression like quantitative loci. What they are regions near the gene. Sorry, I'm forgetting the name of it here, but they're regions that are near genes that affect whether the genes are being expressed or not. And what I've done here is I should have mentioned this, that the red one here shows the mutations that strictly fall within those regions. But when I allow some flanking regions upstream and downstream of the genes, I've included these into it. Now, using EQTL analysis is sort of is things to start considering for where you can go with this. But then you'd need gene expression data and then start looking. So that, yes, it's an interesting case. It's definitely worth considering. There's two. I think your hand was first.
So what's the ratio between the SNPs you find in the coding versus the non-coding?
Right, that's a good. I have got the numbers down somewhere. There was still several. I think about there were 14,000 that met all of the QC procedures. 14,000 of the index SNPs that made it into the SNP study, of which I chose the best 5,000. And I think that there were about 5,000 again that were SNPs that were actually within these coding regions. And then you've got to remember that some of the gene sets, a lot of those are larger and there's a lot of duplication that goes on within the genes and the gene sets. So SNPs can feature in different ones.
Then the coding ones, does it change
No, I see what you mean. No, I wasn't actually drilling down because you can have with these mutations. I didn't know how too much to go onto this, but you can have what's called synonymous or non-synonymous mutations, where you can have some where if it appears in a coding region, if that happens, you'll actually get a different amino acid chain that goes into the protein. That information just says. What I wanted to do here was really compare it with the polygenic score analysis, which also doesn't take into account of these things. But these are all things that are worth doing for future research. Yes, there was a question there.
Are there any effects related to sex or race in this kind of disease, and then also in SNPs and so on? Because
Okay.
I'm for sure that in SNP they have like the differences, right?
Right, they do. There is no effect of race in this because, how do you say this in a PC manner? The different effect, what they call population stratification, you do have to only include people from a same ethnic population that goes into this, and these were Caucasian Brits that went into this, because there are distributions of different mutations amongst different populations. So if you were to have, for example, all of your cases come from a Chinese population and the controls come from white, you would see amazing significance, but of course you'd be measuring. You can actually, when you do analysis like this, you can detect differences between races. The gender thing, the reason why I haven't looked at it in this data set is there was a lack of information in it. There is actually another data set with far more people it's very recently been genotyped, and it's the last sort of, I'm going to replicate that on that, and I'm going to look at see if there's a gender effect, because one idea is that being female can be slightly protective against schizophrenia. So would it be the case that females need a larger sort of genetic risk load to get the disorder? That's what we're going to consider. So that's like going to be the last push in the last 6 months. We'll consider that question.
Yes, so projecting phenotype directly from your genotype, it always fraught with difficulties, because as you allude to,
Absolutely, absolutely.
As far as I see, you've got a very exciting way of improving the prior, which then allows you to improve whether you go ahead and do more applied analysis of active gene expression.
Right, okay.
The problem being, of course, is that epigenetic factors and other parts that affect expression of genes and all that sort of matter come from a much, much larger part. The other problem you have, of course, is that schizophrenia is very, very hard to model in animals. It's very, very hard to make schizophrenic rats.
Well, yeah, it's a cognitive. It's cognitive disorder. How do you mean behavioural traits?
Like making a depressed rat is a bit easier, but making a schizophrenic rat is particularly difficult. So it's whether you think there will be any ways, if there are anything you can see of improving, because of course you probably have to take some brain tissue in order to look at active gene expression in an effective manner, and whether there's any scope, because at the moment this looks like that would improve how much you would have to do active gene profiling in order to get sensible results about aetiology.
Okay, can I talk to you afterwards about that? That sounds interesting. So you did have another question. I remember you were just going to ask something else.
Yeah, actually,
And I'll get back.
just like curiosity. The first thing is, what kind of technology did you use to get the mutations? If it was like
To get the what, sorry?
What technology did you use to get the mutations? So it was like all genomes.
They were all on the ones that I'm using here were on 2 chips. There was an Illumina chip and an Omni chip that we used for that. I did initially keep them separate because I wanted to use one as a test data set and also to see whether there would be any sort of batch effects from the different chips. Turns out that I could use them together. That's why I've combined them. That's what happens when you talk to. That's the question. Was that answer? Is that okay?
The other thing it was, did you consider about epigenetics? So if there is anything
Yeah.
that
It is a big thing that's going to come into it, and it's all across the whole fit. You know, that it comes into it. But because the thing is, I will get to it. This that you know, if you have using DNAs to predict it, you know, we acknowledge that there are problems. For example, if you have an identical twin that shares your DNA, I think it's about 50% probability that you're going to develop the disorder as well. That's the sort of level of risk. So even with even if you've got identical, it's not deterministic amongst those that have it. So it is a very difficult area. So Giles.
That leads into my question, which is, what do you know about environmental factors? Have you got any data on, say, cannabis usage? I've got a friend with relatively mild schizophrenia who suspect that cannabis, well, certainly might have helped his case.
Right.
So do you have access to any of that?
Not on this data set, because this is the way they get this data for the cases is that these people receive clozapine, and because of the side effects of this drug can be really nasty, so they're constantly monitored for when they're taking it, and they constantly give blood samples. It's these anonymised blood samples that they've agreed to be genotyped, so we can take no other information on these individuals.
So you've got patients here who have the worst type, the least treatable schizophrenia, but another question is, are you sure they've all got the same type, or do you think you're actually trying to classify several things with one classifier?
Well, that's a big problem. Is schizophrenia a single disorder in itself, or is it it's a spectrum of disorders? It's fraught with this. And sorry, what was the question you have? Well, ultimately, the problem that you have with this is, well, the phenotype. Yeah, there's an assumption that, and this was the study that actually used these, is, you know, oh, sorry, I should have actually mentioned this was a study that was carried out by Marion, Hampshire, at Cardiff University, but I'll put that on the slides when I put them out there. But ultimately, the phenotype is they receive clozapine. Now, they've been deemed by psychiatrists to suffer from treatment-resistant schizophrenia. So yes, it is a noisy phenotype, unfortunately, that we're dealing with.
Which machine learning methods did you consider to use, and why did you use SVM?
The reason why I used an SVM was because I think SVM has flexibility with how it builds the decision boundaries. When I started this, I would have liked to have tried the random forest because you can get the feature importance that comes out of it. But I think the way that it breaks down and it makes the dichotomous choice at a feature if it's above or below this feature. And I think the decision boundaries, if you were to look at a random forest, would be more sort of following horizontal and vertical lines. And I just found that, well, when I did that, I was getting receiver operating characteristic scores around 0.5. So I did consider them, but I just found that this one was the most fruitful that I was getting.
You said twin brother has 50% probability of.
It's something like that. I normally just sit around and build machine learning.
Yeah. Is it possible to have data about relatives and comparing those with and those without?
Well,
filtering out.
Well, initially that's how a lot of the work in schizophrenia genetics was carried out. They did what was called linkage analysis. So you would have fewer samples and you'd say, okay, we've got affected families here. So what is it about the mother and the father that's passed on to these infected siblings, or infected siblings, and those that don't get passed on to the non-infected siblings? And there was a lot of research done about that. But then what they realised was, because those sort of studies are really good if you want to find mutations of a large effect. So I think that's how they discovered Huntington's disease, because you do have this mutation in the Huntington gene that if you have it, you will unfortunately go on to develop that disease. But the problem with schizophrenia is that you need such large sample sizes to deal with it. And therefore, once you then say, is it all going to be data? Now, they are looking at sort of large consortium of what they call trio data. So you have got mother and father as well as the child DNA, but I'm not too familiar. There are people in the department that work with this, so yes, it is being done. Unfortunately, as PhDs go, as you further you go along them, they get more and more and more narrow. So you're mentioning people can make all these fantastic things on either side. Yeah, I can, but I can't. I can't do them right now. I'm still going along here.
Such a method could, in some sense, control for common factors which are not.
Okay.
Okay. So
I think that's that. Well, I think so, but then There are people looking into it, so be aware that it is being done in the field. So I've just been told apparently.
No,
And you
it's
guys want to eat
something
lunch,
happening
surely
up
you don't
until
listen to me.
lunch, so you can hold them as long as you can keep it. I'm just
Have
saying
lunch.
It is officially lunchtime. Maybe another round of applause for that fantastic.
Thank you very much.